Dr. Choi, would you like to take over? I think you've got some slides to share.
Sure, absolutely. Thank you, Dr. Sheehan. Dr. Curry and I are really excited to talk to you a little bit today about our project. We have a couple slides here, and I think we're going to split it halfway through just so that we both have a chance to talk a little bit about the science and hopefully answer some questions. But today we'll talk about a project that we've done together at Mass General Brigham surrounding the idea of cell therapy for brain tumors. Just to set the stage, cell therapy is a really burgeoning area of research, growing exponentially. You can see I updated this slide a month ago, and you can see if you actually type in something like chimeric antigen receptor, which many of you guys are familiar with in the clinicaltrials.gov, you can see how many trials have just accumulated over the years and really is exponential. Most of these are in liquid tumor like leukemia and lymphoma. But if you actually go and look at solid tumors, and so if you use that same search terms for cell therapy, things like chimeric antigen receptor, T cell receptor, or tumor infiltrating lymphocyte, and then you put in that Boolean AND gate of carcinoma to kind of estimate how many of these things are for solid tumor. Solid tumor studies are growing really rapidly as well. And I'd like to show this slide just because this is over 20 years ago. This was got me interested in the field of tumor immunology. Dr. Steve Rosenberg at the NCI was showing this paper back in 2002 of fungating melanoma on the back of this gentleman's neck. At that time, they were taking the tumor out surgically, taking a part of it out, growing out the T cells that were naturally in those tumors, growing them in a certain mixture of cytokines, et cetera, and infusing those T cells back in the body. And they were, at that time, 20 years ago, getting really robust responses against these cancers. But I mean, until it wasn't until 2024 in February that this was an FDA-approved therapy. So it's the speed of a little bit about the speed of science, but also that this form of therapy has been promising for some time. This is a patient that we just took care of recently at Mass General Brigham. It's a gentleman with actually metastatic melanoma, and he had failed checkpoint therapy, had failed small molecule inhibition, and the oncologist came to me and said, "Hey, what can we do here for this patient?" Had a left frontal metastatic lesion, and I said, "Well, why don't we try actually taking tumor infiltrating lymphocytes out of that tumor in the brain and grow them out and infuse them back into people?" And actually, before we did this, we didn't know if this would actually work. We knew that you could take tumors from the periphery and make these cells. We didn't know if we could do this from the brain. So I did a left frontal awake craniotomy on this patient. We took out the tumor, grew out the lymphocytes here depicted in green, and we were able to see not only regression of the tumor at the tumor location, but even at sites that were remote in the brain on the contralateral side in the periventricular area here. This gentleman is about 6 months out from that and is doing very well. And this was like his last line, maybe fourth line of therapy that we had to provide. But we've known for some time that metastatic lesions are different than gliomas. So unlike mets, gliomas have T cells, but they're few and far between. You can see here is a photomicrograph of a glioma. These individual red dots indicate the T cells, and we think that T cells are an important part of the immune response. They're there, but they're few and far between. And so one way around that is to create more tumor reactive T cells. And we can do that with CAR T cell technology, where we take an artificial molecule like a chimeric antigen receptor, and we put it in a T cell. We put that in a T cell to allow it to recognize any tumor target of interest. And these are translated in tandem with co-stimulatory domains that activate the T cell in the tumor microenvironment. This is what it looks like clinically. We can take out T cells. We usually modify them genetically using a lentivirus in our case, but you can use other methods. Expand those T cells 100,000-fold ex vivo and then reinfuse them into the body. And you've actually seen several articles published over the past few years recently about how these CAR T cells can be used in other solid tumor settings, not just liquid cancer. So GD2 CAR T cells in the setting of high-risk relapse refractory neuroblastoma, even brainstem lesions here on the right in work led by Michelle Manji and colleagues at Stanford, and similar tumors out of Seattle with Nick Vitanza and Mike Jensen. But over the past year or so, we've actually seen this mini explosion of CAR T cells for glioblastoma, which we all know is a very difficult cancer to treat. And so a couple of different institutions published papers back to back showing that this might be a feasible way to treat these tumors on the left-hand side, and work led by our colleagues at Penn, Steve Bagley and Dr. Don O'Rourke. And on the right, we have Christine Brown at the City of Hope. And on the bottom right is our paper that we published in the New England Journal of Medicine, where we showed that we treated our first 3 patients with this CAR team platform in the setting of recurrent glioblastoma, and I'll talk a little bit about that today. The target that we target with the CAR team platform is EGFRV3, GFRV3 primarily, which, as you know, is a tumor-specific mutation of the epidermal growth factor receptor. The first CARs to actually target V3 were developed and first clinically trialed in 2017 by Donna O'Rourke and Marcelo Mas at Penn. And this was a study that they performed. They infused several patients peripherally with EGFRV3 targeted CAR T cells, and they learned a couple things from that trial. Ultimately, tumors progressed, but they found out that the CAR T cells could make their way to the tumor, that they could actually target EGFRV3 effectively in the tumor, but that this was actually accompanied by robust regulatory T cell infiltration and upregulation of PD-L1, as you can see in the bottom left-hand corner, which were proposed reasons why we didn't see sustained anti-tumor activity with that therapy. So we went back to the drawing board and we actually tried to develop new CAR T cells to address those things that we saw in that initial clinical trial. For instance, I noted the upregulation of PD-L1. Well, we went to the lab and we said, well, what if we took a CAR T cell that targets EGFRV3 and then CRISPR out the PD-1 surface antigen? So these CAR T cells are impervious to regulation by PD-L1. We did that. And another question was, well, if you just target EGFRV3, what happens if the tumor recurs just after targeting one antigen? So we created what we call tandem CAR T cells that can target two antigens at once, in this case, V3 and IL-13 receptor alpha 2, which is another frequently commonly expressed glioma antigen. For many reasons, we actually just settled on this approach, which is the CAR team approach. This is a chimeric antigen receptor that is designed against EGFRV3. It homes the T cell to that microenvironment, and then it releases a biologic called a T cell engaging antibody molecule, which is depicted here. It's a bispecific antibody that binds to wild type EGFR, which is more broadly expressed at the tumor site, and redirects it against T cells. So this allows the molecule to actually harness even untransduced, unmodified T cells in the microenvironment and redirect them towards the wild type receptor, which is more globally expressed compared to EGFRV3. And the thought was that since you use these CAR T cells as kind of like a micropharmacy, you're able to target something like wild type EGFR without creating systemic toxicity to even healthy tissues that might express the antigen, because it's just released at the tumor site. And so this is what it looks like kind of microscopically. The CAR T cells are depicted in red. They're engineered to express M-cherry. They release these bispecific antibodies, and these BiTEs, or the CAR teams rather, they're synonymous words for the same thing, are able to redirect not only CAR T cells, but even bystanding cells that aren't transduced with the CAR. So they turn basically any T cell in the environment into a tumor killer. We showed that the T cell engaging antibody molecules can actually redirect regulatory T cells in the microenvironment, and ultimately studied in several preclinical models where we implanted human tumors into the brains of immunodeficient mice and treated these mice with, and showed that actually the CAR team approach could actually treat even heterogeneous tumors, or tumors that only had EGFRV3 in a portion of the tumor. Lastly, we actually showed that on mice that were engrafted with human skin on the dorsum, that we were able to safely actually avoid immune reactivity to healthy human skin or healthy human tissues that express EGFR at a physiological level, because the EGFR targeting only happened within the brain. So that led us to actually open up our first in human clinical trial. Dr. Curry is the PI of this study. I think he's going to take it from here, talk a little bit about the science, about how we actually translated this into our first few patients that were treated, and introduce one of our patients who's actually still alive and doing well. So Dr. Curry, I'll give it over to you.
Yeah, thank you, Dr. Choi. And thanks to the Seattle Science Foundation and the Journal of Neuro-Oncology for having us here at Tumor Talk today. It's really a great honor. And as Dr. Choi just described, a tremendous amount of preclinical work went into designing and understanding the activity of the CAR team construct, and including the safety work that was done up front. And that really allowed us to go right ahead into this phase 1 clinical study. Furthermore, this is on the backing of the phase 1 style, looking at CAR T cells targeting EGFRV3 that Dr. Mouse and Dr. O'Rourke had done previously, published in 2017. So we did go ahead with this, what I would say is kind of a standard phase 1 open label study with a plan of treating up to 21 patients, all of which would get treatment. And we did divide these patients into 3 basic arms based on MGMT methylation status and also whether or not the tumors expressed EGFR V3. And I'll go into that in a little bit. First, we treated 3 patients, go back 1, in what we call the safety run-in with a single intraventricular injection of the product of CAR-TME without lymphodepleting chemo. And particularly in other studies, like in hematologic malignancies, often CAR-T treatment is preceded by lymphodepleting chemo. And so we did publish the results from the safety run-in, and the rest of the study is currently ongoing. These patients are now actually getting lymphodepleting chemo and then being treated multiply with the CAR-TME construct. Arms 1 and 3 are currently open, and those are for patients with recurrent glioblastoma, whether V3 positive or V3 negative. And that's based on the legitimacy of treating V3 negative patients is actually based on our preclinical data, which showed that we had impact even if the patients were V3 negative, presumably because of the T-cell engaging antibody molecule. And then arm 2, which we haven't actually gotten to yet, is treating upfront newly diagnosed patients with the known V3 mutation. Next slide. And this is the basic clinical trial strategy. So patients will undergo initial resection and treated with a standard of care, the Stupp protocol. And then at some point they recur, at which time we enroll them and then typically re-resect or at least biopsy so that we can get an understanding of the V3 status and the EGFR amplification status at that time. And then we also place an Ommaya reservoir at that point. And this is all actually after we've done a leukapheresis. Patient recovers from surgery in the usual way, and as long as they remain eligible, and they all have, we infuse them with CAR T cells through the Ommaya, and then we follow response with a lot of actually progression, survival, safety, and also generous correlatives. And these are the 3 patients published in the safety run-in, and you can see they're all kind of typical characteristics. By coincidence, all the patients had tumors in their dominant hemisphere, actually. These were all IDH wild type patients. You see the methylation status, and that refers to the status at the time of recurrence. You see the variable EGFR data. I think really interesting, all 3 patients were V3 positive at the time of original presentation. However, patient 3, subject 3, had lost the V3 mutation by the time we treated them. And they were all treated with 10 million, single infusions of 10 million cells. Next slide. And generally, for these 3 patients, across the 3, we would see very early regression of the contrast-enhancing tumor within 1 or 2 weeks. And you see the examples there. And no dose-limiting toxicities. Patients often had a very early fever, which crested near the end of the first week. And you see per patient, patients 1, 2, and 3, the radiographic changes that we saw within a few days. For instance, you see the patient 1, that response was within 1 day of infusion. Patient 2 had a very durable, has had a very durable response out to more than 3 months, and certainly has a RANO described partial response. And you see the patient 3 data, dramatic reduction in enhancement, mass effect, and even edema just at 5 days after that infusion. Next slide. And patient 2 is kind of our poster child patient of this run-in. He's there in the blue T-shirt. We can use his name. There's an article about him in New York Magazine with a lot of detail about his experience and his long-term survival. He continues to survive without progression at this point. I'm going to turn it over back to Dr. Choi to go over some of the correlatives, which we're really excited about, which kind of gives a little bit of a clue as to what's happening and also how we might be able to follow progress.
Thank you. Yeah, I'm going to just pick it up here. I think an important thing to remember is each of our patients has an Ommaya reservoir with a ventricular catheter placed. into the lateral ventricle. And so that actually affords us the opportunity to pull off CSF as well on regular intervals to perform correlative studies. And in this case, this is one of those studies that we looked at. Basically, we looked at extracellular vesicle-derived RNA corresponding to both EGFRV3 and EGFR copy number. These are the targets that we're actually going after with our therapy. And so you can see that in many of the patients early on in the therapy, they had elevated levels, but then these decreased over time to almost undetectable levels. In addition, we were able to actually look at in the CSF things like vector copy number and different cytokines. I mentioned before, and Dr. Curry mentioned that this was a very relatively tolerated treatment where patients did not have any dose-limiting toxicities, but we look at things like cytokine release syndrome, ICANS, and TIAN, and these are all grading scales used to actually kind of capture neurological toxicity from cell therapies. Nothing over grade 2, nothing sustained outside of 2 weeks. We did manage fevers with a drug called anakinra, but none of the patients required steroids in the immediate post-infusion period. They're all monitored on the floor. They're not in the ICU, and they go home after several weeks of inpatient monitoring. You can see that vector copy number for the CAR T cells is elevated over time, but it decreases towards 28 days. And so after a month after infusion, you don't see as many of these cells. In terms of interferon gamma, TNF alpha, interleukin 6, these all peak within the first 7 days of infusion and then decrease to almost baseline levels after that. Interestingly enough, if you look in the peripheral blood of these patients, you can actually detect CAR T cells. They get out. We infuse them in the CSF, they get out into the periphery. You can see spikes in some patients at about 2 to 3 weeks after infusion. You can see them. But interestingly enough, if you actually look at the CAR T cells in the periphery, only about 0 to 2% of those CAR T cells are actually positive for the T cell engaging antibody molecule, meaning that they don't have it bound to their surface and they're not reactive to EGFR wild type. Whereas if you look in the CSF, 70% of the CAR T cells actually have detectable bispecific antibodies. So it's kind of implying that this is working the way we want it to. That within the anatomic compartment of the CNS, we can actually get the dual targeting to EGFRV3 and wild type, but in the periphery, it's otherwise safe. And over time, we've had the opportunity and pleasure of working closely with Dr. Moss to actually create several in-house investigator-initiated homegrown constructs that we make in the lab and then offer to patients at Mass General Brigham in the form of phase 1 clinical trials. This is just a handful of these novel CAR T cells that we've actually been able to produce in the laboratory and then translate early on. And this is obviously none of this work is done in isolation. We have a huge multidisciplinary team. It takes a little army to actually complete these types of studies. Obviously, Dr. Curry is here. He's the PI of the study, but along with Elizabeth Gerster and myself are investigators on the study, and Marcella Moss is the sponsor of the study, along with a whole host of other individuals who help with the correlative studies and also the inpatient care of these patients who can get relatively sick from the infusion, but ultimately, as I said, nothing was dose limiting. So with that, I'd like to just close. I think that's our last slide. Thank you all for your attention and allowing us some time to present us our data and share it with you. And thanks for that opportunity. I know that we'd both be happy to stay on and answer some questions.
Fantastic presentation and obviously incredible work. I think if there's ever an optimistic approach that could lead to paradigm shifts with glioblastoma, this is certainly shows tremendous promise. Chris, I think you had a question.
Yeah, I've got a couple, and I'd also invite our audience that's watching in real time, if they have any questions, to please put them in the chat feature, and we're happy to ask them in real time. But thank you both for joining us and for giving a great talk. I've been following the CAR T program at Penn for a number of years, actually almost 15 years now. And the question I have for you is a bit of a downer. It comes from the practical side of this and the cost limitations. And I know from the heme malignancy side, they were ready for prime time. And then a lot of centers found it to be a cost prohibitive endeavor for patients. So where do you see how much development needs to go beyond what's already been done to make this a practically feasible endeavor for patients outside of trial?
Maybe I can take a stab at that, and Dr. Curry, if you have any thoughts. I think you bring up really great point in this. I think with any kind of emerging technology, we run into the balance between cost and efficacy and commercialization and ultimately scalability. I think we are still in the phase of like, is this a feasible way to treat these patients? And can we get to a point where we can prove that? But even I mean, I think you're getting at the right point, is that even to get through a phase 2 or a phase 3 to do this, the amount of money or funds or resources needed required to actually support that study or definitive or registrational study is quite a hurdle. You know, we are looking at different ways to have off-the-shelf therapies that can be given in a non-autologous manner. I think this is going to be a huge way forward. There are other strategies that use NK cells or cells that don't require an autologous format, as we mentioned. But I think our hope is that with the ease, you know, when we started this, the idea of a closed system, the Prodigy system being available at 1 or 2 places was a dream. Now you see them popping up everywhere. Our hope is that the technology becomes cheaper over time and we're able to provide it in a more expeditious fashion. But it's a tough question. I think it's a real challenge for the field.
Yeah, I'll just add, it's the real cost, or the kind of exorbitant, unmanageable cost, really comes in the development, in my opinion. Like Bryan was saying, phase 2, or imagine a randomized phase 3 study, how many patients you have to enroll and treat and do controls with. I do think that once you're able, a lot of the cost is not just, it's the infrastructure you need for the autologous cell preparation. A lot of the cost is actually in the creation of vector to deliver the construct to those CAR T cells. That was actually a very surprising cost for me. But I think that with maybe regionalization and proper investment in infrastructure, you know, you'll never see this done at every community hospital, or maybe even every academic medical center. But this is like the question for all of gene and cell therapy. And if you think that gene and cell therapy is part of the future of the treatment of disease, which I think is reasonable, we're going to have to figure out how to manage that. So that's like our society, industry, government are all going to have to invest in it.
Yeah, thanks. And the majority of the patients had already received radiation therapy, correct? These were all recurrent ones that you were looking at. What's your thought about the blood tumor barrier, the blood-brain barrier in those patients who already received RT? And do you think that if you ever got this to newly diagnosed patients, would there have to be a way to at least disrupt some of that to be able to get the CAR T cells to where they need to be? Because really, you're throwing it in the ocean and somehow it's ending up in a little stream somewhere in the middle of a landmass, basically. So you know, although they're targeted, you still have to get them from the CSF space to the tumor, right? So do you think there's some element of disruption that's there from both surgical intervention previously, radiotherapy, and how would you is there any other additive therapies you think might benefit getting the cells to where they need to be?
Yeah, and if I may interject there too, great question, Chris, and I would throw in just something that's been of interest to me, which is the application of focus ultrasound. Is there potentially a value proposition there for focus ultrasound to maybe lead these to a more targeted approach, particularly if one thinks, as Dr. Curry had suggested, maybe the EGFR V3 status is not quite as super relevant, or if one were to think about a sort of a general potential CAR T approach that may have that really may have multi-target CARs or bispecific CARs that could almost be a little bit more off the shelf and then made for that individual's T cells. Could we think about use of focus ultrasound and maybe some multi-targeted CARs that would work in a typical recurrent glioblastoma patient?
I think one of the short answers is potentially that when we started this whole journey, we felt that T cells, unlike kind of macromolecules or other biologics, were living, and they could somehow find their way. We know that they get through a even intact blood-brain barrier in kind of settings of neuroinflammatory disease. But the point taken is that maybe access is still not as good as it could be. So I don't think that the door is closed on combinatorial therapies. I think focused ultrasound is a really growing area of research that we still understand so little about what it not only does to the blood-brain barrier, but to the microenvironment in general. And I think that goes probably along one of the largest or the heaviest burdens that we face with a targeted therapy like a CAR T cell, is the fact that these tumors are just infiltrated with so many counterproductive immune cell types. And we mentioned that the idea of maybe treating newly diagnosed disease might even be better, because some of those tumors aren't as infiltrated and aren't as harsh in terms of their environment. So things that modulate the microenvironment and otherwise, taking that into consideration with clinical trial design is certainly, I think, in the next phase of what we're considering.
And I'll just supplement, but actually, just based on the observation of watching brain tumor immunotherapy for a while, there are ways to increase the number of T cells in tumors, whether it's by vaccine or otherwise, or we've seen it with the CAR T studies, including the 3 that we referenced earlier in the talk. But it's just this. So I don't think it's a blood-brain barrier, but it's actually a challenge for all of solid tumors, so even like peripheral non-CNS solid tumors. It's one of the things keeping it from being as successful as in hematologic malignancies is A, the heterogeneity, and B, actually just trafficking into the tumors. All of which is a way of saying that another approach like focused ultrasound actually could be very, very attractive. And going through the work of timing that out is something we're really interested in.
Well, truly, such a hypothesis generating presentation, and a very impressive set of results. Looking forward to hearing more, and I hope you'll come back and talk to us. And obviously going after Nature and New England Journal and other papers are fantastic. I'm so honored that you were able to put some of this forth in J&O, and come back anytime, submit anything. Love to have updates from you and your team. So I'll channel my Tumor Talk colleague and say that we usually try to keep this pithy and have the content usually be about 30 minutes long. Surgeons' attentions are usually 30 minutes or less in many instances. So with that, we'll conclude. Thanks so much for joining. This will be archived on the Seattle Science Foundation for those who are interested as well. Thank you.
Fantastic.
Thank
Great to
you.
be here. Thank you.
Thanks so much.
Bye-bye.